Hello, and welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Today we'll be talking to Dr. Brian Diers about stacking soybean cyst nematode resistances via plant breeding. What are nematodes? How do we combine resistances? How can we tell if it worked? Answers to all these and more coming right up. I'm your host, Abby Morrison. Let's talk about science. Hi, Brian. Welcome to the show. How are you doing today?
I'm doing well, thank you.
Excellent. We'll just launch right in here. Why don't you tell us a little bit about yourself, your background, and how you came to your science?
Yeah, so I'm a professor at the University of Illinois in the crop sciences department. So I originally grew up on a farm in Minnesota, so I've had an interest in agriculture my entire life. When I started at the University of Minnesota, I really didn't know what I wanted to do, but just finally was drawn to crop science, largely because I took a botany class that I found very interesting. And then I took a genetics class, which I really enjoyed. And plant breeding and crop science was a way that I could put these two interests together. So after I did my undergraduate at the University of Minnesota, I then went to Iowa State, where I did my master's and PhD program, both in soybean breeding and genetics. I then spent a year as a postdoc at the University of Wisconsin, actually working on brassicas. And then I spent about 5 years at Michigan State as an assistant professor, and then finally came down to the University of Illinois, where I've been for about the last 20 years as a professor in the department.
So we're talking about soybean cyst nematodes today. Can you tell me a little bit more about what that is?
So soybean cyst nematodes are a nematode pest, and nematodes are microscopic worms. So they're very small worms that live in the soil. We're concerned with soybean cyst nematodes because they feed on the soybean roots. And when they do that, they reduce yields. So as I said, soybean cyst nematodes are microscopic. So we can't actually see them with the naked eye in most stages. We can, however, see the nematodes when the pregnant females emerge from the roots. And they're really basically kind of at the limit of what you can see visually. So when they come out of the roots, you can just barely see them as kind of little white specks on the roots. Although they're very small, they can really cause a large amount of damage. So if we have high infestations, we can see, 30, 40% yield losses. In places like Illinois, often you don't visually see the problem, although it can be reducing yields. Part of that is that most of the symptoms are below the ground. So, the nematodes are feeding on the plants below ground. When we have good soils like we have in Illinois and other places in the Midwest, we can't really see much in the way of visual symptoms from them, but still we're losing yield because of it.
You've got 3 papers published with us. We're going to focus on one of them today, but can you tell me some of your other work that you've done in the past with your other 2 papers?
Yeah, so we've been very interested in studying the impact of soybean cyst nematode on soybean production. We've been very interested in understanding resistance to soybean cyst nematode. So one of the things we did a couple of years ago, we published a paper where we looked back at data across, I think it was the last 11 years of some regional testing that we had coordinated. And what we were able to show was that at actually pretty low levels of SCN infestations, we could see an advantage of having genetic resistance to soybean cyst nematode. One thing is that we looked at also different sources of resistance, and in typical fields, we weren't able to show advantages of one source of resistance over others, but in heavily infested fields with nematodes that can overcome our major source of resistance, we could show that some of the alternative sources of resistance actually were very beneficial.
And what about your 3rd paper?
So one of the things that we're very interested in doing is identifying alternative sources of soybean cyst nematode resistance to try to broaden the base of what genes we're using to confer that resistance. So what we had done was we many years ago, identified a wild soybeans, and the species is Glycine soja, accession that showed resistance to soybean cyst nematode. We know that soybean was domesticated in China by early farmers a few thousand years ago. And so what happened was that Glycine soja is a very viney plant that has tiny black seeds, and the pods open up right when the pods mature, so the seeds shatter on the ground. The early farmers, through just generations of selection over thousands of years, basically domesticated soybeans into what we see as soybeans today. So we went back and we identified this Glycine soja line that showed resistance to soybean cyst nematode. Through some collaborations, we were able to map two major genes from that Glycine soja line that conferred resistance. Through breeding, what we were able to do was go through generations of backcrossing to basically move those genes into a background that is suitable for growing in the Midwestern U.S. And then in this third paper, what we did was painstakingly fine map the locations of those genes. So what we did was we developed populations segregating for the genes. We then identified recombinations close to those genes. And by doing that, we're able to identify the precise locations of the genes. So why do we need to do that? Well, one reason is that breeders want to be able to use genetic markers to select for soybean cyst nematode resistance. It's actually a very difficult trait for us to actually phenotype. You have to grow the plants in a greenhouse, you have to inoculate them with nematodes, you have to grow them for 30 days, and after 30 days pull up the roots and count how many female nematodes have been produced on those roots, which is something that's, takes a lot of time and effort. What we prefer to do is be able to isolate DNA from individual plants, test them with genetic markers, and use that information to predict whether or not the plant has a resistance gene or not. And by precisely or fine mapping the locations of these genes, that allows us to have very good markers that are very close to the genes that gives us an accurate prediction whether or not a plant has that gene or not. Another reason for actually fine mapping the genes is that what we want to do is to clone these genes, meaning that we want to identify the DNA sequence of the gene that actually confers the resistance, because that helps us better understand how this resistance works and perhaps in the future improve on the resistance we have.
Wow, that's I had no idea about any of that. That's great. Moving on to the paper we want to focus on today, can you give us some background on this one, what your goals were in this experiment, what your hypothesis was?
Yeah, so one of the big issues that we have in the Midwestern U.S. with soybean cyst nematodes is that farmers have access to varieties that have resistance, but the vast majority of these varieties, probably somewhere around 95% of the varieties, all have resistance from the same source. And this source is PI 88788. This was actually a session that's in the USDA Soybean Germplasm Collection here at the University of Illinois that was collected in the 1930s by some USDA plant explorers in northern China. They collected this accession when nematodes were first identified in the U.S. Some researchers, I think it was back in the 50s, started screening the collection for resistance, and this was a line that was identified as having resistance. And breeders have been very successful in incorporating this resistance into high-yielding varieties. But as you can imagine, we don't want to depend on just one source of resistance, because what we can see over time is that the nematodes in the fields are becoming more and more capable of overcoming this resistance. Therefore, as we look into the future, we know that this resistance is eventually going to break down and become ineffective. So what we're doing is looking at alternative sources of resistance, and we're trying to combine genes from various sources to see whether or not by taking, let's say, the genes I talked about from Glycine soja together with the genes from PI 88788 and another source, can we get much higher levels of resistance that will be durable in the longer term.
And we talked about this on the phone a little bit, but can you talk about how these resistances stack? I mean, are they kind of morphing together to create something new? Are they just separate, stacking on top of each other?
Yeah, so what we do through breeding, basically, we'll combine these different genes together by crossing and selection. And we know these genes are all located at different places on chromosomes. So we know we can combine these all together. How they actually all function together when they're combined into one variety, we don't really understand that very well. And if anything, it just looks like when we combine genes together, we get additive effects, meaning, let's say if one nematode resistance gene increases resistance by 30%, we add the second gene that adds another 30%, so therefore we would have, let's say, 60% resistance or something like that. Beyond that, we really don't understand how these combinations really work together.
I imagine it's a bit of an art too, because I would guess it's maybe not exactly, 30 and 30. Do you find that that's the case, that they interact in unexpected ways?
I mean, they can do that. So for example, a major SCN resistance gene that has been used by breeders that comes out of 88788, actually it's at the same locus as that, but actually from a different source. We know that that actually interacts with another gene, and you need those two genes together to get full resistance. But how that interaction works, we don't understand. The research that we published where we combined the two genes from glycine soja together with the 88788 resistance combined with the gene from the third source, we didn't see a lot of interaction. It looked more like it was an additive improvement of resistance.
Can you tell me more about how you actually completed this research, kind of the methods you used?
Yeah, so we spent initially a long time just getting our gene combinations together. So as I mentioned, what we did was we took resistance from PI 88788, and we know that that's largely conferred by a single gene called RHG1. We then had two genes from glycine soja that we had worked with. And then we had another gene from a third plant introduction. Third PI is PI 567516C. Right? So we had those three different sources. So basically we made crosses between them. We selected a plant that was heterozygous for all four genes. And from that, we then developed a population of soybean plants that would have different combinations of these genes. We're able to do this because we have good genetic markers for all of these genes, so that we could go into a large population of plants, test them with markers, and say, yes, we know this plant is homozygous for, let's say, all four genes. This plant has two of the resistance genes, but not the other two, et cetera, et cetera. So we're able to classify all the plants for what genes they have. Once we do that, then we're able to take the progeny from those plants and test them for SCN resistance or soybean cyst nematode resistance. And we do that in a greenhouse. So what we do is we'll plant the seeds in little PVC tubes, and those are suspended in crocks in a water bath so that we can control the temperature of the roots. And then we inoculate those plants with eggs of soybean cyst nematode. We grow the plants for 30 days. We then pull the plants out of the tubes, count how many female nematodes emerge from those roots, and that tells us the level of resistance each plant has. So then what we're able to do is correlate the information we know about what genes each plant has versus how many nematodes are on the roots, and that tells us basically how these genes are working in terms of providing resistance. What we did then was we did this for a number of different strains of nematodes, and what we really wanted to do was understand how these gene combinations will work with strains that we know can really overcome some of the resistance we already have in varieties.
I've got a couple questions for you out of this. First, when you have these genetic markers, how do you check to see if they have that marker?
This marker work is largely done using a process called polymerase chain reaction. And how polymerase chain reaction works is that basically we're able to amplify specific sequences of DNA, right? So we know where these genes are located, and we know DNA sequences around those genes, and we're able to amplify the DNA close to those genes, analyze that DNA to determine whether or not the resistance gene is there or the alternative gene for susceptibility is there.
So you use the fine mapping to figure out where these genes live on these chromosomes, and then you use the PCR to zoom in and see if it's there?
That's right. So we can use the PCR to look at these populations of plants and figure out which plant has what gene. Again, it's based on linkage between these genetic markers and the gene itself. But by doing the fine mapping, we know that our markers are very, very close to the gene, so that the predictions are highly accurate.
The second question out of that is when you're looking for resistance, if there's 30% and 30%, how do you get those percentages? Is it, we expose them to approximately this many nematodes and we found this many survived, or is it by plant survival?
Yeah, so what you do is you inoculate the plants with a known number of nematodes. And in our tests, we used about 2,000 nematode eggs per plant. And basically, the nematode eggs are suspended in a liquid solution, and you basically pipette it into each tube where the plants are. And then at the end of the experiment, you count how many females are produced. And what we do is we always calculate the level of resistance based on a susceptible check that's included in the experiment. So basically, we'll say it has 30% as many nematodes as the susceptible check, or 80% or 0% as a susceptible check. And that allows you to compare results from various studies, because the environment during these nematode trials will influence how much reproduction of the nematodes you actually have. But by putting it all relative to a susceptible check, we can still make comparisons across studies.
And is 2,000 eggs pretty standard for what you would find in an average field, or would that be considered like a really infested field?
Yeah, so it's hard to compare. When you actually look at fields, what you do is you look at how many eggs per 100 cc of soil. And if you had 2,000 eggs per 100 cc of soil, that would be pretty heavily infested. And I, quite honestly, we would probably have probably a little over 100 cc of soil in those tubes, but I'd have to actually do that calculation. Yeah, it would be pretty heavily infested.
Sure.
How about that?
And then I wanted to ask you, before we move on, about the work environment. I know we touched on this on the phone. Illinois is kind of a big center as far as having these different seed varieties, right?
Yeah, so the University of Illinois actually houses the USDA Soybean Germplasm Collection. So USDA maintains germplasm collections for major crops. So there's like a collection for corn, collection for wheat, et cetera, et cetera. So we have a soybean germplasm collection, which is housed here. So there's a USDA curator who maintains a collection, and he has staff here that maintains this collection. This collection contains over 20,000 different soybean types. So these would be, let's say, old varieties. These could be just land races that were grown by farmers decades ago in China and places like that, as well as it could include wild soybeans, the glycine soja that I talked about before, which is a source of a couple of these resistance genes we're working with. So anyway, this collection is a tremendous resource for soybean researchers, because one thing is that soybean, when we go back and look at the pedigrees of our current varieties, although there's hundreds of varieties available to farmers, they all trace back basically to a handful of introductions that were brought into the U.S. in the early 1900s. So we don't have a lot of diversity within soybeans. And so one of the things is that we can use the diversity in the collection that's maintained there to actually increase diversity and bring in new genes that are useful for the crop.
That's great. It's used obviously for research, is it also used as a historical archive, like the seed arcs, or do they use it to try and introduce new species for eating or the other uses of soybean?
Yeah, so it's used for lots of purposes. So source of new genes. If if you're looking for, let's say, an old variety, it can be a source for that. One thing that we actually did, and we published a paper in Crop Science a few years ago, where we actually went back and we looked to see how soybeans have improved over the last 80 years of breeding in the U.S. And we were able to go back to the collection and actually obtain varieties that were grown all the way back to the 1920s.
Wow.
By doing that and by comparing directly the performance of these old varieties and current varieties, we're able to show how we've actually changed soybeans over time through breeding.
That's amazing.
Yeah, so there's a lot of uses for this collection, but the main reason is to just have this as a repository for genes that we can use for improving soybeans. Let's say we get a new pest or something like that. We can go to that collection to see, is there resistance to that pest in some of this germplasm?
Sure. And then final question related to this. I'm so fascinated by it. Can you describe what it looks like in there? Is it a mixture of seeds, live plants, just a forest?
Yeah, so the collection, the good thing about soybeans is that we can preserve the soybeans as seeds. And I think they can keep seeds for about 10 years. And then they need to regrow the plants to produce fresh seed. So really the collection itself would be a cold room that contains lots of small boxes that would have the seed from the 20,000 plus different accessions. Also, every year what they'll do is they'll grow a fraction of the collection out in the field to produce new seed. And so the collection would also include these field grow outs where they grow the plants, double check to be sure that the plants are correct, and then they'll harvest the seed, make sure the seed is correct, and then they'll put the fresh seed back into the collection. Another thing with the collection is that it's also backed up in Fort Collins, Colorado. So it's important, we don't want just a collection here, let's say a tornado hits the building where all the seed is housed, we wouldn't want to lose everything.
Right.
So everything is backed up at Fort Collins. I think a lot of it is also backed up at the gene bank that's in Norway. And you've probably heard about that in the popular press, where they built this gene bank basically into the permafrost in Norway. And a lot of the accessions from the soybean collection has been sent there as well as a backup.
That's great. That's so interesting to me. Hey everybody, I hope you're enjoying today's episode. Interested in learning more? Brian's article, Pyramiding of Alleles from Multiple Sources Increases the Resistance of Soybean to Highly Virulent Soybean Cyst Nematode Isolates, published in Crop Science, will be freely available for the next 2 weeks on our digital library. You can find a link to it in our show notes. Let's get back to the show. So moving on, tell me about your results. What did you find?
So what we found in general was that by combining genes from these different sources, we could get higher levels of resistance. It's dependent on the SCN isolate that we used, but in general, what we showed was that if you combine genes from, let's say, these 4 different sources, we can get much higher level of resistance than we can if we only use one source. So what we're thinking about is, what can breeders do in the future to get better resistance, especially being that this 88788 resistance is breaking down. And what I think this shows the direction that by taking these genes from glycine soja, from this other PI, combining it together with the RHG1 from 88788, we can get overall a better resistance package. And one thing we've been doing is we're working with Melissa Mitchum, who's a nematologist at the University of Missouri, and she's actually been looking at how we can use these different gene combinations actually in rotations. And she's been doing some greenhouse tests looking at different rotations, and one thing she observed was that if we have the combination of genes that I just talked about, and we rotate that with an alternative source of resistance, we're actually able to slow down, it appears, the reproduction of the nematodes over generations of selection. So, I mean, I think this could be a useful thing for breeders to do, to think about, let's take these genes from these different sources, combine them together to get a better level of resistance.
Yeah, for sure. Can you tell me why it's so crucial that research like this is being done now?
Yeah, so it's important for this research to be done now because we don't want to wait until our resistance doesn't work at all before we start on these kinds of things, because the breeding process actually takes quite a while. So let's say our resistance no longer works in the fields, it'll probably take us 8 years before we can get new sources of resistance bred into soybean varieties.
Wow.
You know, in many ways, what we need to do is prepare ourselves for the future when we know our current resistant sources are not going to work very well. And again, it's because this process just takes a lot of time, and you can't make extremely quick progress.
So as far as future research goes, where are you hoping to go next?
Yeah, so the thing that we want to do is be able to get our results into the fields of farmers, obviously. I went into plant breeding because I want to do things that are useful for growers. So we've been working very hard in breeding these genes, especially from glycine soja, into good, high-performing backgrounds that have competitive yield in Illinois and other places in the Midwest. And we recently released a variety that actually has those glycine soja genes in it. But we're going to continue working on this, and we're also working with other breeders so that we can get these genes into the hands of other breeders as well. We've done also some studies to look at what kind of yield benefit these genes have, and what we're able to show is at least if we grow them in locations where there's no nematodes or very low levels of nematodes, there's no yield drag associated with these genes. And in places where there's high nematode populations, we can show a yield advantage. So again, what we want to do is just get these genes into good yielding backgrounds so farmers will have choice in sources of resistance for the varieties that they're going to be growing on their farms. One thing I'd like to be sure is that we can acknowledge the funding sources.
Okay.
So funding for this work and a lot of the soybean research that's been done in the public sector is paid for by the Soybean Checkoff program. And what happens is that when soybean farmers sell their seed, 0.5% of the value of the sale goes into the Soybean Checkoff program. And what happens is that this funding or this money is then administered by committees of farmers who decide what to do with it. And part of that money goes into research, and that's been very helpful, and it's allowed us to do the research we're doing and a lot of other research going on in the public sector.
Great. I've got 3 questions left for you. First off, if people want to learn more about SCN resistance or your research or anything like that, where can they go to learn more?
So if you want more information about soybean cyst nematode, you certainly could look at the articles that we've published that has the information that we've just talked about. But I would also suggest going to your state extension websites. And for example, the University of Illinois, Iowa State, other extension organizations will have lots of information on soybean cyst nematodes. I think you can probably find what you're looking for at these places.
Second question I have for you. If people want to get involved with this or help somehow, what are some ways that they can do that?
So what I would recommend is that farmers, what they really need to do is know more about the nematodes in their fields. So for example, you really need to take soil samples. You need to see what is the nematode infestation levels in your fields that you're growing soybeans. And if you see over time your infestation levels increasing, that suggests that actually the nematodes are able to overcome the resistance you're using, assuming you're using resistant varieties. And what you need to really do is be informed about the sources of resistance in your variety. So when you buy the seed, you need to ask the seed salesman, what's the source of resistance? What are some alternatives, especially if you're finding that the nematode population is increasing in your field? You know, if you're growing a resistant variety that has resistance from PI 88788, you might ask, you know, what other sources of resistance do you have available so that you can rotate your resistance sources.
Final question for you. What is one fun fact people wouldn't know about you if all they had was your research?
One interesting thing that I'm working on right now is that I'm involved in a USAID project where I'm involved with working with soybean breeders in Africa. And, you know, one of the problems in Africa is that there's a real deficit of protein on the continent. And soybeans is a great source of protein. And so what we're doing is we're working with some soybean breeders there to help them develop soybean varieties that can help smallholder farmers in Africa so that they can improve their nutrition. They can also produce some soybeans that they can sell to help pay for schooling for their children and things like that. So it's been a really interesting project for me to work on because it's given me a whole new insight into, you know, limitations for agriculture.
Yeah, that's great. That sounds very rewarding.
Yeah, it is. It really is.
Well, thank you so much for being on the show today. That's all the questions I have. Thank you so much for your time. Thanks for being on the show.
You're welcome. Thank you.
Thank you for listening to Field Lab Earth. You'll find a link to today's paper and other resources for this episode in our show notes or on our website. If you have any questions, comments, or recommendations for show topics, please contact us at podcast@sciencesocieties.org or on Twitter @FieldLabEarth. If you'd like to hear more content like this, please subscribe, and don't forget to rate and review us on iTunes or anywhere else you find your podcast if you like our show. This podcast is a joint production of the Tri-Societies, the American Society of Agronomy, Crop Science Society of America, and Soil Science Society of America. Special thanks to Lobo Loco for the use of their song Spook Castle on the intro and outro of our show. Opinions and conclusions expressed by authors are their own and are not considered as those of the American Society of Agronomy, Crop Science Society of America, Soil Science Society of America, its staff, its members, or its advertisers.